Welcome to part 2 of the lecture on chapter 7, microbial genetics. So in the first part, we talked about DNA structure, DNA replication, DNA transcription, and DNA translation. Essentially the process that allows us to get from the DNA molecule that holds the genetic information, or multiple molecules that hold the genetic information, all of which are called the genome, to actually accessing and then making a functional unit off of a specific gene. Now, of course, this process needs to be highly regulated so that you only utilize the genes that you need at a specific time within a specific environment. That's really obvious in multicellular organisms because we have different types of cells that are actually very, very different in their structure and their function. But remember that those cells all have the same genetic information as each other. So the cells in our eyes have the same genetic information as the cells in our skin, but they are expressing different genes, right? And even at the bacterial level, where you have a very much simpler organism, or a prokaryotic level, you still have to be able to regulate which genes are being used at which time. So for example, if there is a specific metabolite in the environment, being able to turn on the genes that allow you to metabolize that specific sugar or molecule, and then turn it off when that molecule is no longer available, or that nutrient is no longer available, right? So that you're not wasting resources. And it's a common theme in biology that living systems need to be efficient in the use of their resources, because resources, energy, and nutrients tend to be limited. And so we will do a brief discussion about the regulation of genes. We'll basically use a couple of examples that are really prominent in prokaryotic systems, so we can kind of see some examples of how systems are being regulated, these genetic systems are being regulated. Okay. You'll remember in our previous discussion that we said that sometimes genes are copied as a single individual gene, and sometimes they're copies as groups of genes called operons. And when we were completing that worksheet, you noticed that eukaryotes actually have what are called exons and introns, and they will actually copy a group of genes and then kind of modify which genes they want to utilize. Prokaryotes don't do that, but they still do sometimes copy out, or oftentimes copy out, groups of genes that are together called an operon. And so when we talk about these two examples, the lac operon and the trip operon, we're talking about a group or a cluster of genes that are all related to one another and tend to be copied out and then utilized together. Okay. So here's an example where you have a long strand of DNA in the original genome that has various regulatory genes ahead of it. So those genes will allow you to turn it on or off in terms of copying that genetic information. And then the operon itself that has a promoter that can then promote the various other processes that we need to copy it out into a protein. And then the actual genes are a series of genes that are there. Okay. So we have two kind of examples of how genes can be regulated. First of all, before we get to that, genetic expression can be regulated at the transcription stage or the translation stage. And so this first example here is regulating genetic information at the transcription stage, where you're actually, whether or not you make a copy of the DNA in the first place. And then after you make that mRNA copy, then there's a second set of stage or set of tools that the cell can use to actually activate or deactivate an mRNA copy as to whether it will be translated into a protein or not. So this first example is talking about turning on or off the actual transcription of a gene. And then we'll talk in a minute about turning on or off the translation of a gene or a series of genes. So essentially, there's kind of two examples. One is called inducible operons, and this is genetic information that's usually dormant. It's usually not active, because oftentimes it's related to metabolism. And so if those nutrients that are needed for metabolism aren't there, then why should you code that region? So usually it's dormant, but there are ways in which you can turn on that region or that operon. Repressible operons are the opposite. They're genes that are usually active because they're important in production of, membrane proteins, for example, or various types of amino acids, things that are needed all the time within the cell. But sometimes if you have an outside source of, or you have enough of those macromolecules, then you can actually just shut off that operon. And so the examples that we'll use are the lac operon, which is related to the metabolism of lactose, and the tryptophan operon, or trip operon, which is related to the production of the amino acid tryptophan. Okay. So with the lac operon is usually turned off in prokaryotes. It's not utilized continuously, and the reason for that is that lactose is a sugar that can be used to extract energy from, but glucose is a much more efficient energy source. And so lactose is not going to be the first go-to for a prokaryotic organism or for other types of organisms as well. And so having that gene kind of consistently off and then only turning it on when you need it. There's two examples of when you might need the lac operon, or when a prokaryote might need the lac operon. The first example is in the case where you had really low levels of glucose, right? So there's not enough glucose around, so you have to turn to other sugars. And that's what you're seeing here, and I'll explain it to you in a second. The second example is when there's a lot of lactose around. And so if there's a lot of lactose around, why not metabolize it, right? Even if there's also glucose around. And so this first example is when we have low levels of glucose. And actually, when levels of glucose become very low, a related molecule called cyclic AMP, or cyclic adenosine monophosphate, will become high in quantity. So there'll be a lot of cyclic AMP around. Okay, and that indicates to the cell that there's not enough glucose around. And so cyclic AMP will actually then bind to a promoter site ahead of the lactose operon, the lac operon, on the DNA. So what you're looking at here is DNA, and those are the actual genes. And ahead of the genes, there is this site called the CAP site, which is the catabolite activator protein site. And if it's activated by the cyclic AMP, which indicates that glucose levels are low, then the lac operon is transcribed. And then you actually make the genes that you need to actually metabolize lactose. Okay, so that's the first instance of turning on the lac operon, is when glucose levels are low. The other potential time when we would need the lac operon, or the prokaryote would need the lac operon, is if there's a lot of lactose around. Let's utilize it, right? And so there's another way of inducing the lac operon, is that we have this repressor protein that tends to be bound ahead of the lac operon, just at the beginning, right around the promoter of the actual gene, or a series of genes. And when that repressor is bound, you will not have transcription of the gene. But in the presence of lactose, there is that lactose will be turned into an associated molecule, and that molecule will actually bind to the repressor. And once it's bound to the repressor, that's it. It can't be bound. The repressor molecule, which is this, or protein, which is this right here, can no longer be bound to the promoter and can no longer inhibit the transcription of that operon. And so in that case, again, you remove the off switch on this lac operon, and therefore you do actually transcribe the lac operon, and then you can make those proteins that you need to actually break down lactose. And so in this case, the reason why you do that is because it's indicated by these molecules here that lactose is present in the environment and it should be utilized. Okay, and so that's the example of the lac operon, where we can see how at the transcription level, the cell is actually deciding whether or not to make functional or transcribe a gene that is usually turned off. The other example that we have is called the trip operon. And the trip operon, just get to the trip operon here. The trip operon is the group of genes that allow you to make the amino acid tryptophan. And in E. coli, generally speaking, tryptophan is needed. Amino acids are really important because they're continuously being used to make proteins, right? And so the tryptophan operon, or set of genes that allow us to make tryptophan or allow the prokaryote to make tryptophan, are usually activated. But there are times when the organism can actually get tryptophan from its environment or from the nutrients that it intakes. And in that case, it doesn't necessarily need to make its own. So it won't spend the energy that it uses on making tryptophan. It won't waste that energy. And so what happens with the trip operon is that the operon is usually transcribed. It's usually a functional gene. And so you can see that here. There is the series of genes that are used to make the various parts of tryptophan. And then ahead of that, there's a regulatory gene that can either turn it on or off, but usually it's on. But if in the environment or in the cell you have tryptophan, so these circles are tryptophan, they'll actually bind to another repressor that will actually repress the transcription of tryptophan. And you can see that here.
The TRP operon of E. coli includes a promoter, an operator, and 5 structural genes encoding proteins involved in the biosynthesis of tryptophan. The TRP operon is a repressible operon, meaning that the genes are usually transcribed. When excess tryptophan is available to the cell, the operon is repressed, ending the production of the proteins of tryptophan biosynthesis. The E. coli chromosome contains a regulatory gene near the TRP operon. This gene is constantly transcribed and translated to produce repressor proteins. However, these repressor proteins are inactive unless excess tryptophan is available. Once activated by tryptophan, the repressor proteins can bind to the TRP operator, preventing RNA polymerase from moving beyond the promoter. When a repressor protein is bound, the genes for tryptophan biosynthesis cannot be transcribed into mRNA. When more tryptophan is available to the cell than the cell can use, some tryptophan molecules bind to and activate the repressor proteins. The repressor proteins bind to the operator, preventing the transcription of the structural genes that encode the proteins of tryptophan biosynthesis. This regulation prevents the cell from producing excess tryptophan when tryptophan is already available. When excess tryptophan is not available, the repressor proteins are inactive. The structural genes of the TRP operon are transcribed and translated, producing the proteins necessary for tryptophan biosynthesis.
Okay, so that gives us an idea of how genes can be regulated upon transcription. Okay. Now, after you've already transcribed a gene and you have an mRNA strand, you can also still regulate the translation of that strand. And there are a number of different ways. And most of them involve another RNA molecule that then interacts with that RNA, with that mRNA, and then turns it off, essentially. That can be temporary or, completely turning it off or breaking it down. So we have microRNAs, miRNAs, and these are small segments that bind to the RNA region, and sometimes they even cut the mRNA. If they cut it, that's it. That mRNA will no longer ever be translated. You also have small interfering RNAs, and they're another kind of RNA that can bind to an mRNA and then just, inactivate it. Now, there's also something called a riboswitch. Sometimes riboswitches are on mRNAs themselves. Sometimes riboswitches are on a secondary RNA molecule. But what that is essentially a piece of the RNA molecule that's folded under certain conditions and unfolded under other conditions, right? The most prominent, or one example, is the plague pathogen Yersinia pestis. And this actually has its pathogenic or virulent genes are turned off by a riboswitch until it's under the about 37 degrees Celsius temperature, which is the temperature of our bodies, which we're the host. Humans are the host. And so once it changes temperature from an environmental temperature to 37 degrees Celsius, then that riboswitch actually changes shape and then allows for those virulent genes to be translated. Okay. And so these are ways in which the genes can be regulated at the translation step. Okay. So the next discussion we want to have is on mutations. So go ahead and review gene regulation and then prepare for the discussion on mutations. Again, you can add terms in your note file that's in the description as well. Okay, so surprisingly, with all of the complexity that we've described today, genes are usually copied and even transcribed and translated efficiently. Okay, but specifically when we're talking about mutations, we're talking about the copying or the replication of genes. So kind of back to the beginning when we were talking about those various processes. And so when we're talking about replication of the DNA, there's usually only about one mistake in every 10 million genes that are replicated. Okay, so our cellular structures are very, very efficient in replicating our DNA. But it is possible that a mistake is made. And the mistake, if it's made, can affect the production of that gene or that protein, or it may not. It depends on the type of mistake that is made, and it depends on the ramifications of that mistake. So we're going to go back to that sentence that we had used earlier in chapter 7. So if we kind of think of the code in the DNA as we're replicating it as a series of letters, THECATATERAT, right? And that means to us the cat ate rat. There's two types of overall just like big mistakes that you can make. Substitutions, where you substitute one of the letters, in this case the R, for the original C. So there's a mistake in which base pair was added, but there was a base pair that was added. And so in some cases a substitution still makes sense, right? Because if we go back to the amino acids, sometimes the same amino acid is coded for by a couple of different codons. Or maybe different amino acids have similar shapes. And if it's a different amino acid with a similar shape, then it doesn't impact the overall structure of the, eventually after transcription and translation, the protein that is made. Some substitutions make a really, an issue because that new, series of base pairs codes then later in translation for an amino acid that actually disrupts the structure of the protein. That's less likely with a substitution. Oftentimes, the more deleterious mutations are what we call frame shift mutations. And that's where you have a deletion or an insertion that throws off the whole frame, because we're reading in threes. So if you change if you delete or add, then you've actually shifted the whole kind of reading of those base pairs thereafter. And so if we were to add in an R, not switch out an R, but add in an R, now the rest of this sentence doesn't make sense. The cat rat era What does that mean? Right? And so if we're imagining this sentence as a series of amino acids later on, after we've transcribed and translated that portion of the DNA, then all of those amino acids thereafter are then changed. And then oftentimes that protein is not functional. And so these types of mutations can often be even fatal to a cell, but deleterious definitely to a cell. And then there are very few exceptions where mutations are beneficial, very few. And there are exceptions where mutations are just like they don't make a difference essentially. And that is essentially how you build up diversity over evolutionary time, is these small mutations that survive because they're not extremely deleterious to the cell. And then you get different eye colors, for example, and so on and so forth. Okay. And so in terms of, and so we would call that where a silent mutation, this should be silent, not silence, a silent mutation, a missense mutation, or a nonsense mutation. Silent is when you just don't even see it. Missense is where it makes a difference that then makes that, makes a change. But it doesn't impact the overall structure. And nonsense is when that's it. The rest of the polypeptide, it makes no sense and can't really essentially be a functional protein. There's a discussion in your book on how cells then repair DNA after mutations. We're not going to focus on that in this chapter, and you're not going to be tested on it, but you're welcome to read that information as well, because it's beneficial. Now, we said in the normal processes of the cell, mutations are very rare, but there are environmental conditions that will increase the likelihood of mutations by decreasing the efficiency of these processes that we've talked about. And so, intuitively, we know about these various types of chemicals and radiation and, within our systems and within our diets that will then lead to increased likelihood of mutations. Then your body will fix and repair whatever it can. But if the mutagen is too significant, then you will actually see negative ramifications of those mutations, like cancers and other types of inefficiencies throughout the cell and throughout the body. Okay. Okay. So that's our discussion of regulation and mutation. The next discussion we want to have is on genetic recombination. Okay. So oftentimes when we think of genetic information, we think of genetic information being passed down from parent to daughter. But prokaryotes are unique in that they can actually pass their genes horizontally from themselves to peers. And so we want to look a little bit into that. One, because it's interesting in understanding prokaryotic systems, and two, because we actually can utilize those same techniques and methods within the lab, and we can actually move genes between different organisms to create or to generate different characteristics, which we'll actually do in the lab. Okay? And so when we're talking about horizontal meaning between living peers of prokaryotes, versus vertical, which is from the parent to the child, transfer of genetic information. Essentially what's happening is a piece of the genetic information, a couple of genes, is formed into a plasmid, and that's just a small circle of DNA, and somehow that gets passed to a peer. There are kind of three ways that this can happen. Either through transformation, where that plasmid is actually picked up from the environment, or transduction, where a virus is the vector that actually moves that plasmid, or bacterial conjugation, where you actually have a pilus. Remember when we talked about cellular anatomy, prokaryotic anatomy, we said that there are filaments called pili, and an individual pilus can be made to connect two cells, and they can actually transfer genetic information that way. And so you can see here the different processes. First is transformation. Here, what you're looking at is bacterial cells that cause infection and disease in mice. Okay, so these are cells that have that virulence gene. Here, we have cells those cells, but they're dead, right? It's bacterial cells that cause a fatal infection in a mouse. If you kill those cells, then that mouse should not be infected. Okay. Then we have this other strain of the same bacteria or a similar bacteria that does not have virulence genes, and that should not kill the mouse. But if you kill this virulent bacteria and add it to the mouse, inject it in the mouse at the same time as you add this non-virulent strain, then oftentimes the non-virulent strain, depending on which type of bacteria, some are able to do this transformation process and some aren't, will actually pick up the virulence gene from the environment, from the dead cells, and become virulent and actually kill that mouse. Okay, and so they're just picking up that genetic information from around them. Here you have transduction. Transduction, you'll recognize this because of viruses, our discussion on viruses, where the virus is actually bringing in a piece of genetic information. In its normal virulent state, it would actually take over the cell and then make the cell produce more virions. But sometimes it'll insert its genetic information, remember the lysogenic stage, and it will actually just incorporate that genetic information into the DNA of the bacterium. In that case, you have actually incorporated that new gene into the bacterium, whatever it is, and it's now it's called transduction, and it's now incorporated into that genetic information. And then this final example, which we said was bacterial conjugation, is where you have a pilus that's formed. We said that usually there's not very many pili, there's just one or two that are needed to transfer specific genes or plasmids when we talked about prokaryotic anatomy. And so then that plasmid is transferred from this initial cell into its peer. And then that peer now has that genetic information. Now, I want you to listen to something that Jung said in his book that we had read from before, I Contain Multitudes, about this, just to give you an idea of what's going on. When you were born, you inherited half of your genes from your mother and half from your father. That's your lot. Those inherited bits of DNA will remain with you for all of your life with no further additions or omissions. You can't have any of my genes, I can't have any of yours. But imagine a different world. Imagine a world where friends and colleagues could swap genes at will. If your boss has a gene that makes her resistant to various viruses, you could borrow it. If your child has a gene that puts him at risk of disease, you can swap it out for a healthier version. If distant relatives have a gene that allows them to better digest certain foods, they're yours. In this world, genes aren't just heirlooms to be passed on vertically from one generation to the next, but commodities to be traded horizontally from one individual to another. This is exactly the world that some bacteria live in. They can exchange DNA as easily as we might exchange phone numbers or money or ideas. Sometimes they'll sidle up to one another, create a physical link, and shuttle bits of DNA across, their equivalent of sex. They can also scrounge up discarded bits of DNA in their environment, left by their decaying and dead neighbors. They can even rely on viruses to move the genes from one cell to another. DNA flows freely between these types of bacteria. Even closely related strains of bacteria might have substantial genetic differences. Bacteria have been carrying out these horizontal gene transfers, or HGTs for short, for billions of years, but it wasn't until the 1920s that we first realized it. And then he goes on to talk about some of that research, but I'm going to come back to this part here. This process can instantly change microbes from harmless gut residents into disease-causing monsters, from peaceful jackals into sinister hides. They can also transform vulnerable pathogens that are easy to kill into nightmarish superbugs that shrug off even our most potent medicines. The spread of these antibiotic-resistant bacteria is undoubtedly one of the greatest public health threats of the 21st century, and it is a testament to the unbridled power of horizontal gene transfer. And so I read that to you with some slight modifications because he over-exaggerates that all bacteria can do this, and it is some bacteria that can do this. So the genetic information that can be transferred between bacteria, it's brilliant because you have all of this kind of sharing of different information. But from a medical perspective, it can be very dangerous because you can actually spread antibiotic resistance much more quickly throughout a population or throughout a community, not even a population, not just one species. Sometimes you can actually transfer genes between related species. And that can spread very quickly, much more quickly if we were just relying on horizontal gene transfer. And so being aware of the importance of horizontal gene transfer, as we had just mentioned it, where prokaryotes are sharing genetic information, is really important. Now, another thing that makes horizontal gene transfer interesting is because we can actually utilize it in the lab. And so even bacteria that don't tend to pick up genetic information, we can actually use various techniques, shock them essentially, into actually being able to pick up genetic information. And so we can use modifications of these same similar techniques that bacteria are using naturally to move genes from one species to another, even from different domains to another, right? You can have plants genes being moved into bacteria. And so you can actually transfer different phenotypic characteristics by moving genetic information around via these bacterial processes. And so in the lab, we're actually going to move a gene from a jellyfish that allows jellyfish to glow in the dark into a bacterium and actually change that bacterium to actually be able to phenotypically express that gene and glow in the dark. Okay, and so that's one of the procedures that we will do, and that is called recombinant gene procedures or technology, and that is used. That is mentioned at the end of your book. You can read over that section and also read over the final lab that we'll do, lab 10. Will give you more information on how we utilize these techniques in the lab. So that's our discussion for chapter 7. We talked about, in the first part, we talked about the structure of DNA replication, transcription, and translation. In the second part, we talked about regulation of those processes that we had talked about in the first part, and how bacteria have some unique characteristics around gene transfer and mitigation. So feel free to go ahead and jump back into your notes packet, finish up all of that information. That'll help you solidify chapter 7 and also study for the exam for this class. And then email me if you have any questions. So thank you for listening, and I will see you in the next lecture.